Good evening, everybody. Welcome to the seventh session of the life problem solving as interactive session. We will be starting in a couple of minutes if people join in, if some more people join in. Mm. Mm. Okay, I think we have waited long enough. Good evening. Once again, welcome to the seventh session of the live interactive problem solving session of the NPTEL course, Genome Editing and Engineering. This course is taken by Professor Utpal Bora from IIT Guwahati. And I'm Sanket Mukherjee, a Prime Minister's Research Fellow from the Kusuma School of Biological Sciences, IIT Delhi. So we will go on directly into the questions. So the term clustered regularly interspaced palindromic repeats was coined by A. Alexander Bolotin, B. Francisco Mojica, C. Barango, or D. Andrei Markov. I'll give a minute for everybody to think on this, and then we'll discuss the answer. Okay, so the answer to this question will be Francisco Mojica. So the term clustered regularly interspaced palindromic repeats, or CRISPR, was coined by Francisco Mojica. So let's talk about a little bit of history of CRISPR. So in 1987, while investigating the alkaline phosphatase isoenzyme conversion mechanism in Escherichia coli, Ishino and his group discovered a loci containing a series of 5 repeated palindromic sequences in the 3' end flanking region downstream of the IAP gene. The repeat sequences were separated by variable sequences of uniform length with no homology to each other. Although of no relevance to the focus of their work, they nevertheless dedicatedly reported about this unusual sequence structure with an unknown biological function in their conclusion. Thereupon, it was soon forgotten by the scientific community. Um, so in 1992, Francisco Mojica at the University of Alicante in Spain, while reviewing genome sequence data from the extremely halophilic microbe Halopharax mediterranei, noticed 14 unusual DNA sequences, each of which are 30 bases long. Each sequence read roughly the same backwards and forwards, and they repeated every 35 bases or so. Now, Mojica had to struggle for funds to pursue research on his this new discovery that he had made, and interest was even ridiculed by peers and decision makers for his a quest to pursue in these strange little repeats. So at that time, there was not much which was known about repeats or their biological function. So few said that there are many repeats in many organisms. We have known about them for years and still don't know how many of them work, they would say. So they would also discourage him by telling, don't care about repeats so much. At that time, none, including Mojica, knew that these repeats were going to change the course of biotechnology revolution in the coming decade. So the term clustered regularly interspaced palindromic repeats, or CRISPR, was coined by Mojica in correspondence with a team of Dutch scientists led by Rut Janssen at Utrecht University in 2002. Janssen in the same year identified another set of sequences present with CRISPR, which is called CRISPR-associated genes. That is what we call as the Cas gene, and hence the term CRISPR-Cas is always used together. By 2005, Alexander Bolotin found that spacer sequences between the CRISPR sequences shared similarities with the DNA of viruses. They also discovered 2 to 6 base pair long protospacer adjacent motif, a conserved sequence present at the end of the sequence similar to spacers in target. It was hypothesized that CRISPR has some role in immunity of the host bacterium. So before these discoveries, adaptive immunity was thought to be um, a particular feature of higher order organisms, but with introduction of CRISPR, with discovery of CRISPR, adaptive immunity in prokaryotes was also studied again. So we'll talk about it. So how did the timeline worked out? So in the year 1987, discovery of tandem repeats in the sequences of Escherichia coli was found, and in 2002, the name CRISPR was coined. In 2005, spacer sequences in bacteria and archaea were found highly homologous with their viruses, the viruses that infected them. In 2012, in vitro genome editing technology with CRISPR-Cas9 was developed. In 2013, eukaryotic cells were modified by using CRISPR-Cas9. So the system which was used inherently by prokaryotes for their immunity was exploited to even edit eukaryotic sequences. We'll see how that happens. In 2014, the discovery of the precise method to modify genes in eukaryotes with CRISPR-Cas9 was found. In 2016, oncogene mutations were inactivated by using CRISPR-Cas9. Again, a very big discovery. In 2017, CRISPR-Cas9 used to modify the beta globin gene in human embryo. And in 2019, Jiankui jailed for deliberately violating medical regulations and for rashly applying gene editing technology to human-assisted reproductive medicine. Again, these kind of gene editing technologies always go through ethical violations or ethical concerns, I could say. In 2020, Emmanuel Charpentier and Jennifer A. Doudna were awarded a Nobel Prize for the development of a method for genome editing very recently. So they got a Nobel Prize in chemistry. So we'll talk about the next question. The type 2 CRISPR-Cas system is defined by presence of a large endonuclease, that is Cas9, which ranges from A, 1100 to 1800 amino acids, B, 2000 to 2600 amino acids, C, 1010 to 1600 amino acids, or D, 1000 to 1600 amino acids. I'll give a minute for you to think, and then we will discuss the answer. Okay, so the answer to this question would be option D. Uh, so the type 2 CRISPR-Cas system is defined by the presence of a large endonuclease, Cas9, which ranges from 1000 to 1600 amino acids. Let's look at the classes of CRISPR. So the CRISPR-Cas systems, it can be categorized into two broad classes based on the structure and function of Cas protein, which are further divided into subdivided into six types. So class 1 include type 1, 3, and 4, and class 2 include type 2, 5, and 6. We'll see the classes. Type 1, 2, and 5 systems recognize and cleave DNA. Type 6 can edit RNA, whereas type 3 can edit both DNA and RNA. Let's see how it looks like. So as you can see in the figure on the right, type 1. So essentially, all the different types, they have different enzymes that are, Cas proteins that are associated with them. So like type 1, E has Cas7, and then Cas3, Cas1, Cas5, these kind of enzymes. Type 2B is also very similar. Whereas type 3B, type 2A has Cas9, type 5 has Cas12A, type 6 has Cas13A. So these are different proteins, which have different functions in specific. So all CRISPR-Cas systems, they contain Cas1 and Cas2. The three different types, type 1, 2, and 3, are each categorized by unique Cas proteins involved in the maturation of the CRISPR RNAs. targeting of foreign nucleic acid and nucleic acid cleavage. So the type 2 CRISPR-Cas system is defined by presence of a large, around 1000 to 1600 amino acid endonuclease, Cas9. So that's the unique feature for type 2 CRISPR-Cas system. Coming to question number 3, which of the following is not a key step of CRISPR-Cas immunity? Is it option A, adaptation, option B, expression or biogenesis, option C, interference, or option D, reproduction? I'll give a minute for you to think about it, and then we'll discuss the answer. Okay, so the answer to this question would be D, reproduction. Reproduction is never a step of CRISPR-Cas immunity. So let's see how CRISPR-Cas system work usually in a bacterial system. So as you can see in the figure on the right, so whenever a viral DNA is incorporated into bacteria, part of it is actually added to the CRISPR as a CRISPR element. So now this acts as a recognition site for further bacterial infections to be recognized. So Cas9 protein, it works in conjunction with these CRISPR RNA, and it essentially recognizes any future viral infections. And Cas9 has the ability to cleave DNA or RNA, so it causes cleavage. So whenever the same bacterium is infected by the same virus, if it has already encountered that virus before, then it can essentially be immune to it due to this CRISPR-Cas system. That is why the immunity is very similar to that of an adaptive immunity in humans. So it stores information about its encounter with invading viruses by storing a fragment of DNA stolen from that virus itself in the CRISPR loci. In later encounters, the bacteria uses this stored information once again in physical form to counter the attack by the virus. This archive provides immunity to the bacteria against viruses, and it's heritable. So even if one bacteria has a CRISPR sequence, it can also transfer that using conjugation or other methods, vertical or horizontal transfer mechanisms in bacteria. It could also transfer this immune information to other bacteria. That is also something that we should know. Bacteria and archaea with RNA-guided acquired immunity to invasive DNA. So that was the discovery essentially about CRISPR. So the key step which are involved in CRISPR-Cas immunity is adaptation. So adaptation is when a new foreign particle, it's in this case, it's a nucleic acid, it's a DNA or an RNA. When it comes inside the virus, it is inserted into the new spacers of the CRISPR locus. So that is the adaptation step. Next is the expression or biogenesis step, where these CRISPR locus is transcribed to form the CRISPR RNA. And then third is the interference step, where Cas9 protein that comes into picture, and or Cas proteins, they come into picture. They detect any future viral encounters, and they degrade the mobile genetic elements by CRISPR RNA and Cas proteins. So we have already talked about it. So this is how it looks like. So whenever a foreign DNA it comes in, it has a part of it is added to the spacers of in the CRISPR loci, and then it forms the CRISPR transcript. And later, this CRISPR RNA is used as the guide for Cas9 protein to cleave any foreign DNA that the bacteria might encounter. So this is the idea behind CRISPR RNA, CRISPR mediated immunity. So question number 4. Neurodegenerative diseases are characterized by the progressive loss of neurons in the dash and peripheral nervous system and the deposition of proteins with altered physicochemical properties. So is it the heart, the liver, the brain, or the kidney? So I'll give you a minute to think about it, and then we'll discuss the answer. Okay. So let's go with the answer. The answer would be pretty obvious because neurons are all present in brain only. So I think the question itself gives the answer. But let's talk a little bit about neurodegenerative diseases. So a neurodegenerative disease or a disorder is characterized by progressive loss of neurons in the brain and peripheral nervous system, and the deposition of proteins with altered physicochemical properties. So it has been observed that almost all of the neurodegenerative diseases, they have a characteristic feature that is aggregation of proteins, misfolded proteins, mistranslated proteins. So it's pretty much a characteristic feature of any neurodegenerative disease. So if you talk about beta amyloid for Alzheimer's, alpha-synuclein for Parkinson's disease, Huntington protein for Huntington's disease, prion, tau protein for again Alzheimer's, TAR DNA binding protein and fused in sarcoma protein are most common protein that contribute to Alzheimer, Parkinson's, Huntington's, transmissible spongiform encephalopathies, tauopathies, and amyotrophic lateral sclerosis, respectively. And these proteins are used to classify neurodegenerative disorders at the molecular level. So the presence of these proteins as an aggregate in brain essentially helps the clinicians to diagnose which kind of disease the patient might be having. So that's the idea behind this. So if one of the most well-researched neurodegenerative disease is Parkinson's disease. It has also been studied pretty well in terms of aging because it's most of the neurodegenerative diseases, they happen when a person ages. So in terms of developing therapies against these neurodegenerative diseases like Parkinson's disease, there could be two ways. One is a disease modifying therapy that could happen by modifying the platelet-derived growth factors, glial cell line-derived neurotrophic factors, brain-derived neurotrophic factor, and new Turin are these are several disease-modifying targets that can decrease the development of Parkinson's disease. So that could be one way. The another way would be a non-disease modifying therapy, which could be used to target the vascular endothelial growth factor A and cerebral dopamine neurotrophic factor are symptomatic, and they target gamma aminobutyric acid or dopamine synthesis. So essentially what happens in case of Parkinson's disease is the accumulation of alpha-synuclein in the substantia nigra of the brain, which causes the secretion of dopamine to be lowered. So one way which is most common is the non-disease modifying therapy, where dopamine is given. Levodopa is given to the patient to relieve the symptoms. So that is one way. A disease modifying therapy would be a better approach to actually find the root of the problem. So if we see, gene editing is one of the most promising tools to actually uncover the molecular basis of Parkinson's disease and find new therapeutic targets and eventually generate new gene treatments. So upregulation and downregulation of gene expression or selective editing of key genes known to be modified in Parkinson's disease, like the Parkin gene, the GDNF gene, PINK1, and AADC, that is aromatic L-amino acid decarboxylase, can be used to correct defects in molecular pathways related to Parkinson's disease. Gene editing is a viable technique for restoring the activity of important biological pathways that are interrupted and potentially contribute to Parkinson's disease. So as you can see in the figure, uh, uh, what happens in Parkinson's disease is, damage in the mitochondria, increased oxidative stress, because there is an increased level of reactive oxygen species that also happens to damage mitochondria. Autophagy is affected, and that includes, ubiquitin proteasome system that is affected. So proteostasis, or the levels of proteins and protein formation, protein synthesis, protein maintenance, and protein degradation is affected. That causes aggregation of alpha-synuclein in the substantia nigra. And these aggregates form Lewy bodies. They are very characteristic way that how they are present. So the idea behind it is we will deliver proper genes, whatever genes are affected or they are disrupted or mutated in Parkinson's disease, we will correct those mutations and we will deliver those genes into the cell, and we will see how if these diseases are cured in that sense. So let's see at some of the strategies. The enhancement of dopamine synthesis is one of the most common, and even now also it's preferred. It's FDA approved, actually, that dopamine availability is increased by just giving dopamine, the protein, as such. The other strategy could be increase the availability of neurotropic factors and neuromodulation in subthalamic nucleus to stimulate brain regeneration. So that is another strategy. The third strategy could be focusing on the genes that are involved in the mitochondrial pathway and mitophagy. And the fourth strategy could be decreasing the alpha-synuclein synthesis, because if it is lowered, then if synthesis is low, then aggregation will also be low. So which might help in alleviating the effects of modified mitochondrial pathways. So that's all about Parkinson's. So a lot of other neurodegenerative diseases, they also follow the same path. Let's talk about question number 5. Which of the following vector is known for high efficiency of delivery, and it is widely used for the delivery of CRISPR components inside the cells for gene therapy? Is it A, AAV, B, RNP, C, DMD, or D, AADC? So I'll give you a minute to think about it, and then we will discuss the answer. Okay, so the answer to this question of the vector which is known for high efficiency of delivery and is widely used for the delivery of CRISPR components inside the cell for gene therapy is AAV, adeno-associated virus. Let's see about it. So adeno-associated virus vectors are known for high efficiency of delivery, and it is widely used for delivery of CRISPR components inside the cell for gene therapy. We'll see how it looks like and what are the components. So the CRISPR components can be packaged as plasmid DNA, encoding its components, including Cas9 and the guide RNA. So the CRISPR RNA is actually, when it is exploited to be used as for gene editing purpose, it's a guide RNA or gRNA, or it can be delivered as mRNA of Cas9 and gRNA. The challenges associated with adeno-associated virus vector is that it can induce viral toxicity, and the CRISPR components undergoes longevity of expression, resulting into potentially high incidence of off-target effects. So uh, what this means is viruses, when they grow in eukaryotic systems, they can hijack their machinery and they can affect the healthy growth of eukaryotic cells. So sometimes it might induce toxicity. Sometimes cells might die because of this extra pressure of viruses that they have on them. And what happens is when these components they are there for long, when they are expressed for long in cells, sometimes their efficiency, their effectiveness also decreases, and they cause some off-target effects that is not desirable in a normal situation. To avert the virus associated risk, it can also be introduced to target cells via electroporation or nucleofaction or microinjection. So these are physical methods. We'll see how they work. These are physical methods which do not use any virus, but they directly deliver the nuclear material, the nucleic acids basically, or the protein, into the nucleus of the cell. So that's the idea. Microinjection is only suited for ex vivo delivery, while electroporation is also largely used for ex vivo. We'll see what they are. Delivery of the Cas9 protein and guide RNA as ribonucleoprotein complexes has reduced the off-target effects while maintaining editing efficacy owing to its transient expression and rapid clearance in the cell. So it is also very important that when you are introducing any genetic corrections in a cell, it is important that, like any medicine, that needs to be cleared by the system. If you're taking any drug, that needs to be cleared in the system, from the system, through kidneys or other metabolic pathways. It is important that even the CRISPR components, they are also cleared off. Otherwise, they might induce toxicity, they might cause off-target effects. So that's the entire thing. So when we are talking about the delivery mechanisms, as you can see, these are the components which contains the Cas9 mRNA or the Cas9 protein directly, or Cas9 could be delivered as a gene in the plasmid. Or we could also have a stable Cas9 expressing cell line, which has the sole function of producing Cas9. And apart from this, the CRISPR, element is also given. The guide RNA is designed and it is given. We'll talk about it again later in some of the questions. So the delivery method which is preferred is electroporation or injection. Even a liposomal or nanoparticle mediated delivery is pretty much popular, although not used in terms of, anyway, CRISPR is not used in any kind of medicine as such, not approved yet. Conjugation to delivery moieties like cholesterol, cell-penetrating peptides, they can also. I mean, these are all the ways how CRISPR can be inserted into a cell, and then it will be translated in the cytoplasm. So whether it is given as a mRNA or DNA, it needs to be translated in the cytoplasm, and then it will go into the nucleus where it will perform its function of gene editing. So synthetic gRNAs or guide RNAs can be applied to a variety of CRISPR-Cas9 experimental approaches. Dual RNA can be co-delivered with Cas9 mRNA, Cas9 protein, or a Cas9 expression plasmid, or delivered into a stable Cas9 expression cell line. Delivery can be achieved using a variety of methods, resulting in efficient gene editing. So there could be ex vivo delivery, where the delivery happens when the cells are taken from the patient. They are taken outside, they are cultured, and then they are the gene of these cells are corrected, and they are again cultured. They are selected for the corrected cells. They are selected, and then they are transplanted back to the patient. So that's the idea. So ex vivo editing targets cells from the patients that are extracted. These cells are cultured and expanded in vitro. CRISPR components to yield the desired edits are delivered through any of the methods of delivery, such as viral vectors, microinjection, or nanoparticle mediated delivery. The edited cells are selected and expanded and finally reintroduced to the patient. So, and we hope that these edited cells, they will expand in population, and the patient will no longer have the disease. So that's the idea. In case of in vivo delivery, in vivo editing CRISPR components can be systemically delivered via intravenous infusions into the patient. The CRISPR cargo travels through the bloodstream via arteries leading to the target tissue, or locally delivered with injection directly to the target tissue. Once delivered, the edits are facilitated in vivo to provide therapeutic benefits. So one of the problems with this kind of editing approach in vivo delivery is that there are a lot of cells which are in diseased condition, and CRISPR based editing is for now not very efficient. I mean, it has a low efficiency. Not all cells where you will give uh, this CRISPR cargo or components will actually efficiently edit the gene, and they will make the corrections. So those kind of problems, we cannot actually select only the corrected cells and then infuse them. So the efficiency is somewhat lowered. But again, these kind of editing is uh, is preferable at a lower stage of like infancy, in case of blastosis, those kind of things. If we take inner cell mass from there, if we want to edit something there, in vivo delivery approach is more preferable. So the design of single guide RNA is crucial to reduce the dash, as it is the sole guide to control Cas9. Is it A, Duchenne muscular dystrophy, a B, off-target mutation, C, Cas9 variants and orthologs, or D, heterologous nucleases? I'll give you a minute to think about it, and then we'll discuss about the answer. Okay, so the answer to this question would be the design of single guide RNA is crucial to reduce off-target mutations, as it is the sole guide to control Cas9. Let's see. Some of the some facts about single guide RNA. So these are the first approximately 20 nucleotides of the single guide RNA are complementary to the target DNA sequence, where you actually want to introduce a double-stranded break or a nick, whatever, depending upon the Cas9 which you're using, followed by a sequence which is called protospacer adjacent motif, which is typically, its sequence is NGG, and could be any nucleotide, and then GG. So although the targeting specificity of Cas9 is believed to be tightly controlled by above mentioned 20 nucleotide guide sequence of the single guide RNA, and the presence of a protospacer adjacent motif adjacent to the target sequence in the genome, potential off-target cleavage activity could still occur on DNA sequence, with even 3 to 5 base pair mismatches in protospacer adjacent motif, distal part of the single guide RNA guiding sequence. So many studies have demonstrated that different guide RNA structures can affect the cleavage of on-target and off-target sites. Hence, this is very important for this role. The reduction in off-target activity in CRISPR-Cas9 system will provide solid genotype-phenotype correlations, enable faithful interpretation of genome editing data, and facilitate the basic and clinical application of this technology. So let's see the single guide RNA. As you can see, the single guide RNA, it contains a seed region and a non-seed region. So let's talk about it. Crystal structure studies and single molecular DNA curtain experiments suggest that while protospacer adjacent motif site is essential for the initiation of Cas9 binding, the seed sequence corresponding to 3' end of CRISPR RNA, complementary to the recognition sequence directly adjacent to PAM, is also critical for subsequent Cas9 binding and R loop formation and activation of nuclease activity in Cas9. So the single guide RNA comprises the seed sequence and non-seed sequence. We already saw that. Numerous observations have revealed that 10 to 12 base pairs of seed sequence adjacent to the protospacer adjacent motif, that is at the 3' end of the guide RNA, determine Cas9 specificity and is generally more important than the rest of the guide RNA sequences. The seed sequence influences the specificity of Cas9 guide RNA binding through multiple potential mechanisms. This sequence determines the frequency of a seed plus NGG in the genome and controls the effective concentration of Cas9 sgRNA complex. So Cas9 binding or sgRNA abundance and specificity. Urate seeds are likely to reduce uh, the sgRNA abundance and increase specificity, since multiple use in the sequence can induce termination. As you know that stop codons are they contain U and can induce termination of the sgRNA transcription. Generally, the mismatches of the 1 to 5 base pairs at the 5' end of the sgRNA are better tolerated than those at the 3' end. So 3' end is more important for specificity. Single and double mismatches are tolerated to various degrees depending on their position in the guide RNA DNA interface. sgRNAs with exceptionally low or high GC content tend to be less active as reported. So let's talk about another question. Question number 7. Specificity of Cas9 nuclease cannot be further increased by A, choosing target sites with few or no close relatives in the genome, using truncated guide RNAs, employing paired Cas9 nicases instead of nucleases, or dissolving Cas9 FOC1 fusion proteins. I'll give you a minute to think about it and then we'll discuss the answer. Okay, so the specificity of Cas9 nuclease cannot be further increased by dissolving Cas9 FOC1 fusion proteins. It is actually it can be cannot be further increased by utilizing Cas9 FOC1 fusion proteins. Let's see. Before we get into this concept, let's talk a little bit about gene drives. So these are selfish genetic elements that are transmitted to progeny at super Mendelian frequencies. So they have a high probability of transmission that is greater than 50%. So gene drive has potential to eradicate harmful disease causing vectors such as malaria, mosquito Anopheles stephensi, reverse herbicide and pesticide resistance in agriculture, and control destructive invasive species. Gene drive targets to induce a trait that is detrimental to the species, such as sterility, whose population needs to be controlled. That's the idea. So when we talk about gene drive, so to introduce gene drives using RNA guided methods, CRISPR-Cas9 could be used. So the advantages of this is that the targeting flexibility of Cas9, it permits the exclusive selection of target sequences with few potential off targets in the genome. Targeting multiple sites increases the cutting frequency and hinders the evolution of drive-resistant alleles, which must accumulate mutations at all of the sites. The Cas9 nuclease can be quite specific in the sequences that it targets. Fruit flies do not exhibit notable fertility or fitness defects resulting from off-target cutting when both Cas9 nuclease and guide RNAs are expressed in germline. So essentially, when we use some other kind of gene editing technology, like we can use restriction enzymes, we can use basically restriction endonucleases, we can use those kind of approaches. When we use or viral methods might cause the gene drive to not be transmitted, because it's transmissibility is also affected by the reproduction of the organism. So that might be affected. So such kind of problems can be reduced using Cas9, which is again very specific, only edits the uh, target of interest. So the specificity, it can be further increased by choosing target sites with a few or no close relative in the genome using truncated guide RNAs, employing paired Cas9 nicases instead of nucleases, or utilizing Cas9 FOC1 fusion proteins, which would even better, because we do not know Cas9 where it cleaves exactly. So using these mechanisms, we could actually increase the specificity of this CRISPR-Cas system. So these strategies have been used to reduce the off-target mutation rate to borderline undetectable levels. Now let's come to the next question. Inteins or intervening proteins are in frame intervening polypeptides involved in dash by an unique autoprocessing where it excises itself out and form of peptide bonds between two flanking extine or external proteins. So will it be A, DNA splicing, B, protein splicing, C, RNA splicing, or D, enzyme splicing? I'll give you a minute to think about it and then we'll discuss. Okay, so the answer would be protein splicing. So intine are in frame intervening polypeptides involved in protein splicing by an unique autoprocessing where it excises itself out and form of peptide bonds between two flanking external proteins. So let's see how they work. They are actually used to control CRISPR chemically. So small chemical molecules can be used to control the conformational changes in Cas9 protein, which in turn affects its activity. For example, in a strategy where 412 amino acid intine is inserted into two different positions in the Cas9 protein, serine 219 and cysteine 574, in these places it's inserted, it causes inactivation of Cas9. So Cas9 cannot cleave the DNA anymore. So inteins are in-frame intervening polypeptides involved in protein splicing by unique autoprocessing where it excises itself out and form of peptide bonds between two flanking extine. So whenever we add a particular compound like 4-HT, 4-hydroxytamoxifen, also known as afimoxifene, um, a selective estrogen receptor modulator, the intine can be removed via conformational change and a self-cleaving reaction happens. So now the Cas9 protein, it returns to its original conformation and its activity is essentially uh, retained. So this is how it's very similar to that of conditional knockouts where you we have already studied about TET-on and TET-off system. It's very similar to that, where we add a particular modulator which causes protein splicing. Intine is removed and the Cas protein uh, essentially functions, which was not functioning earlier. So if there is any tissue specific expression, tissue specific gene selection, correction, those kind of things can be done in using these methods. So mutation in mutation in betaine aldehyde dehydrogenase OSBADH2 leads to production of A, aroma in rice, B, color in rice, C, bigger size in rice, or D, more carbs in rice. I'll give you a minute to think and then we'll discuss the answer. Okay, so mutation in betaine aldehyde dehydrogenase leads to the production of aroma in rice. So this has been specifically mentioned in one of these slides in the lectures. So the article says creation of novel alleles of fragrance genes OSBADH2 in rice through CRISPR-Cas9 mediated gene editing. So one of the applications which has already been done in agriculture by Ashok Kumar and group in 2020, very recent paper. So mutation in betaine aldehyde dehydrogenase leads to production of aroma in rice. So that particular mutation is introduced by CRISPR-Cas9 system. Um and Here we get the essentially uh, an elite non-aromatic rice variety in case of the mutated ones. There are over 40 commonly used guide RNA designers which fall into one of the following three genres only. Find the option where they don't fall under. So is it A, pattern recognition genre, B, feature rule genre, C, machine learning genre, or D, position finding genre? I'll give you a minute to think and then we'll discuss the answer. Okay, so the answer to this question would be D, position finding genre. The rest of the three are actually used for guide RNA designing. Let's see. So there are over 40 commonly used guide RNA designers which fall into one of the following genres. That could be A, pattern recognition genre. The tools in this genre, they depend on the base pairing rule to determine the guide RNAs. Very simple. We'll discuss about each of them. Feature rule genre, a set of features such as GC content, mismatch, and GRN transcription method is used to filter out the unreliable or unconcerned gRNAs obtained by pattern recognition. The C would be machine learning genre, where in this genre, machine learning algorithms are used to integrate the effects of features and thus more precisely identify the guide RNAs. So let's talk about the pattern recognition genre. So what happens? They rely on the base pairing principle. Tools in this category search for a piece of sequence comprising a short protospacer adjacent motif and around 20 base pair candidate
guide RNA complement the query sequence, that is the target sequence in a specified genome. The fewer mismatches uh, the candidate guide RNA has, the greater on-target possibility it is likely to produce. The specific protospacer adjacent motif should be predefined for its diversity in different CRISPR-Cas systems. Another factor influencing a guide RNA pattern is its transcription methods, in which U6 and T7 promoters respectively require G and GG at the 5' end of guide RNA, because that's how they actually transcribe. Some tools such as CRISPR-seq and FlyCRISPR take it into account, while others such as SS Finder and GT-Scan, these are computational tools, they do not. So these are used for designing guide RNA, and they use this pattern recognition genre. Besides, for individual studies, CRISPR-Flash is able to improve the accuracy by incorporating user-supplied somatic mutation data into pattern matching. So yeah, that's another thing. So let's talk about feature recognition or feature rule, Jonna. In this, the subsequent finding that editing activities vary across different target sites indicates the inherent disparity of some targets in the sensitivity to cleavage, and thus ushers a series of explorations to seek out the key features that influence the targeting efficacy. So the features include GC content of gRNAs or guide RNAs. So if there's a very high or low GC content, then the activity is lowered. Frequency of frame shift mutation it correlates negatively with CRISPR efficacy. Poly-T sequences or poly-U sequences, if we are talking about RNA, they induce termination. We already talked about it because they code for stop codon. Composition of nucleobases involved in Cas binding preference. The presence of protospacer adjacent motif preceding G, and the um, absence of pyrimidines in the last 4 nucleotides of guide RNA spacers are preferred. Exon position, lower efficacy when gRNAs they target the terminal coding exon rather than the earlier exons, and the status of the motif and feature enriched around 10 to 12 nucleotide proximal to protospacer adjacent motif in spacer sequences dubbed seed region associated with pairing process and so on. These are all the features, and the feature rule genre, they take care of these features and they design guide RNA based on that. So tools under feature rule genre always integrate several measurable features with the basic pattern recognition approach to provide more information about candidate guide RNAs and target sites. Users can lay down their own rules to filter out the guide RNAs with poor reliability or of no interest according to feature indexes and the corresponding threshold. For example, Cas designer list putative guide RNAs along with their GC proportions and out-of-frame scores that indicate the frequency of in-frame mutation. Besides, CRISPR-era constructs simple scoring rule by arbitrarily quantifying and weighing the information of GC content, polity motifs, and target locations. So then comes this would be machine learning genre. Sorry for the title. So machine learning genre, given that the weights of multiple features remain uncertain, so machine learning has been used to calculate the mathematical algorithms that systematically integrate features for refining the optimal guide RNA. These models always differ in algorithms and information training data. For example, Deutsch et al. Rule set 1 observed that the depletion rates of guide RNA targeting cell surface markers in mouse and human cells, and attributed them to the intrinsic nucleotide composition of target sequences, which then acted as training data to construct the logistic regression classifier for guide RNA activity prediction. So for any kind of machine learning algorithm, you have a test data set and a training data set. So based on the training data set, the features are weighed, and then the test data set. In the data set, those weighed parameters are used to actually give the guide RNA. That's the idea. So let's come to another question, question 11. Which of the following is not a web tool for genome editing? Is it A, QIIME, B, CHOP chop chop, C, CRISPR, or D, eCRISP? I'll give you a minute to think about it and then we'll discuss the answer. So the answer would be QUIME. I think that's pretty explanatory. You can go through the slides regarding the web tools and computational tools for CRISPR. To analyze query sequences in NGS data, BE Analyzer does not require a full wild type sequence for reference, the type of base editor, the desired base editing window, or T, the non-target DNA sequence. Let's give you a minute to think about it, and we'll talk about the answer. Okay, so the answer would be to analyze query sequences NGS data, B analyzer doesn't require non-target DNA sequences. Of course, it will require the target DNA sequence. So for analyzing NGS data, users need to upload their pair and sample NGS data. As an option, users can additionally upload data from a CRISPR untreated control to compare it with the data from the treated sample. To analyze query sequences in NGS data, B analyzer, it requires a full wild type sequence for reference, the type of base editor, the desired base editing window, and the target DNA sequence. So it won't be non-target, it would be target. So we already talked about it. So let's just go through the questions and the answers just quickly, and then I think we can call this session off. So question number one, just in case somebody missed, the term clustered regulatory interspace palindromic repeats, CRISPR, was coined by Francisco Mojica. Question number two, the type 2 CRISPR-Cas system is defined by the presence of a large endonuclease Cas9, which ranges from 1000 to 1600 amino acids. Question number three, which of the following is not a key step of CRISPR-Cas immunity? And that is reproduction. Then coming to question number four, neurodegenerative diseases are characterized by the progressive loss of neurons in the brain and peripheral nervous system and the deposition of proteins with altered physicochemical properties. Question number five, which of the following vector is known for high efficiency of delivery and is widely used for the delivery of CRISPR components inside the cell for gene therapy? It is adeno-associated vector. Question number six is the design of single guide RNA is crucial to reduce the off-target mutation as it is the sole guide to control Cas9. Question number seven, specificity of Cas9 nuclease cannot be further increased by dissolving Cas9 FOC1 fusion proteins. Question number eight, inteins or intervening proteins are intervening polypeptides involved in protein splicing by unique autoprocessing where it excises itself out and in form of peptide bonds between two flanking extine or external proteins. Question number nine, mutation in beta-ine aldehyde dehydrogenase OSBADH2 leads to the production of aroma in rice. Question number 10, there are over 40 commonly used guide RNA designer which fall into one of the following three genres only. Find the option where they don't fall under, and that is position finding genre. Then coming to question number 11, which of the following is not a web tool for genome editing? It's KIME. option A. And question number 12, to analyze query sequences in NGS data, BE Analyzer does not require the non-target DNA sequence. So that is all for today's discussion. If you have any doubts, you may ask me right now. Uh, we shall be discussing about it. You may turn on your mic and speak. Okay, if there are none, then we are already halfway through the course. I hope you all are enjoying this. And there are only a few weeks left. So if there are no questions further, we will be meeting next week at the same time from 6 to 8 p.m. We'll be meeting for the week 8 of problem solving assignment. So I hope you have a nice time learning about this course. So with your permission, if there are no questions, I think we can end this session. If there are no questions, you may leave the meeting.